and DYNE's Fireside Chat. So we're excited today. We're going to have a little bit of a hybrid presentation and then just a really good discussion. So I hope you've tuned in to learn a lot about DYNE's FORCE platform and their clinical program. So today from the DYNE team, we have Ash Dugar and Molly White. And then also to give us a great primer on the landscape, we have Dr. Richard Finkel, who I will let introduce himself and take it away. And stay tuned for the end. We also have CureDuchenne's Chief Scientific Advisor, Mike Kelly, who will be leading a moderated discussion to really dive in. And we wish we were sitting around a fire, but we are relegated to Zoom. So this is our Fireside Chat, and we really hope you enjoy it. So go ahead, Dr. Finkel. Thanks so much for being here.
Thank you. I appreciate the opportunity to speak about the DYNE program. I'm a pediatric neurologist, and I work at the St. Jude Children's Research Hospital, where I direct the Center for Experimental Neurotherapeutics, and we're heavily engaged in several Duchenne research activities. So let's go to the next slide, and I'll just highlight briefly some of my disclosures. And as you can see here, I've worked with a lot of different pharmaceutical companies on a variety of programs, mainly in Duchenne muscular dystrophy and spinal muscular atrophy, as shown here. So let me turn now to the next slide. And I want to give an overview of Duchenne muscular dystrophy from the point of view of a neurologist who sees these patients in the clinic, starting with a little newborn baby on the left, and working through the life cycle, through infancy and early childhood into the teenage years and into the 20s. And what you see on the top panel are changes in the muscle biopsy, again, from that newborn into the teenage years, showing a fairly uniform pattern on the left, working into a progressively more disturbed looking, or what we call dystrophic type of muscle biopsy, characteristic of Duchenne muscular dystrophy. And with these changes, we see accordingly changes in muscle function, and that's shown in the middle panel, from the little baby who's kicking around and looking totally normal to the little boy who's starting to struggle getting up from the floor, to the 7-year-old boy who's having difficulty running, and to the boy in the teenage years who's lost ambulation and is now confined to a wheelchair. But I'll remind everyone that we anticipate that the boys and young men with Duchenne are spending approximately 2/3 of their lives in the non-ambulant phase of their life. So we need to focus on their arm function and their cardiac and pulmonary, and that's highlighted in that far right panel of the young man who you can see is getting some breathing support. So the other point I want to make here is that even in that little baby to the left-hand panel, there are already changes going on that you can't really see unless you go and look for them. You have to measure things in the blood or in the muscle tissue. So that little baby already has a massively elevated creatine kinase level and also an inflammatory marker called NF-kappa B. So we know that this process has already started shortly after birth and is progressive. So what we're looking for here are opportunities to try to control this progressive pattern of weakness, and that it leads also to breathing failure, heart failure, and premature death. So on the next slide, I want to briefly go through some of the current and emerging treatment opportunities for DMD. And we believe now, from the current standard of care guidelines, that all boys and young men with DMD should be at least tried on glucocorticoid medicine, and that includes prednisone or deflazacort, or a new steroid that's been submitted to the FDA for review called vamorolone. And they all work to slow down the rate of progression and keep these boys and young men walking longer, and also helps control the progression of the cardiac and pulmonary disease. But this is certainly not a cure. So on the second point are the DNA and RNA-directed therapies to increase dystrophin. That's the protein that's deficient in DMD. And I've listed here 5 different strategies: the read-through of the premature stop codons with atalurin, exon skipping, of which there are 4 currently FDA-approved drugs. And I'm going to spend some time specifically on this topic because that's where the DYNE program fits into exon skipping. But just briefly mention that there are other AAV or vector-mediated ways of doing exon skipping, gene replacement therapy, and even emerging strategies for CRISPR, Cas9 gene editing. But I also want to point out that there are drugs in development that are independent of dystrophin, but they complement it. So it works together, such as utrophin upregulation, myostatin inhibition, anti-inflammatory drugs, and muscle stabilizing medicines. So in the future, we envision a patient with DMD as probably being on more than one medication, and that could include a steroid such as prednisone or deflazacort or vamorolone, along with an exon skipping drug, potentially, as well as a dystrophin-independent drug such as a myostatin inhibition. So let's focus a little more on the exon skipping topic on the next slide. So it's estimated that about 80% of patients with Duchenne have a particular variant in their DMD gene, a mutation that is potentially skippable. And we'll go into what we mean by that in a minute. But I've highlighted here in the red circle that nearly 13% of patients with DMD are skippable for exon 51. And some of the others are listed to the right-hand part of this slide for exons 53 and 45, for which there are already FDA-approved drugs, as well as others for which there is intense interest. So ultimately, through having a library of different drugs, we might be able to capture about 80% of our patients with Duchenne. On the next slide, I just want to focus in a little more on this and mention that exon skipping is in fact a proven mechanism to address the genetic basis of DMD, and that the current drugs that are called unconjugated PMOs, and that's the chemical name, they're currently available, and yet they just don't get into muscle very well, which unfortunately limits the dystrophin production and the functional benefit. So the challenges of the next generation of exon skipping drugs that are currently in development are to try to improve the delivery of these ASOs, or antisense oligonucleotides, to get into the muscle, including the diaphragm and the heart. And we need to get it into the muscle fibers first, and then see how much of the dystrophin protein is restored. A third important point is not just getting the ASO in the muscle, but how well is it retained so that it can have a sustained benefit and it can reduce the frequency of dosing and the burden of treatment. Finally, we need to identify how safe are these next generation ASOs. Is there a wide, what we call, safety margin? So that's all really important to the design of these clinical trials, to evaluate both the safety and the benefit from this next generation ASO. So on this next slide, I'm just going to highlight that there in fact is a need for this new generation of ASOs. There's a new chemistry, and you're going to hear more of this from Dr. Dugar about what's special about the DYNE-251 program, and that there's these 3 parts that I've listed here, and you'll hear more about that in a moment. But I want to highlight the potential advantages as it's compared to the currently approved exon 51 skipping ASO. From what I can see from the early, what we call preclinical data, that means information from mice largely, this drug gets into the skeletal and cardiac muscle much, much better than the existing drug. And that's also leading to less frequent dosing schedule, which will be much less of a burden to the little boy with DMD and to his family. There also seems to be a sustained, what we call, pharmacodynamic effect, meaning a restoration of dystrophin protein, along with a favorable safety profile. So collectively, I think that this drug, DYNE-251, has the prospect for more impactful clinical response. So let me turn it over now on the next slide And I'm going to let Dr. Ash Dagar pick it up at this point. You're going to hear more about this specific drug and the drug development program. Thank you very much.
Great. Thank you, Dr. Finkel, for an incredibly informative review of Duchenne, emerging therapies, and differences between various modalities. That was super educational. Thank you. By way of introduction, I'm Ash Dugar, the Global Head of Medical Affairs at DYNE Therapeutics. And with me today is Molly White, the Global Head of Patient Advocacy. First, I want to thank Deb Miller, Mike, and the entire CureDuchenne team for inviting us to this fireside chat to update the Duchenne community on the progress of our Duchenne program, and specifically DYNE-251, our clinical candidate designed to treat patients with Duchenne who are amenable to Exon 51 skipping. As always, it's a true privilege to speak with the Duchenne community. These are our forward-looking statements. Please refer to our website if interested in more details. At DYNE, it all starts with our mission. We're laser focused on delivering life-transforming therapies to patients with serious muscle diseases, with Duchenne being a priority. Our mission is core to who we are and what we do every single day. Sorry, Carrie, can you go back to the previous slide? Thank you. We're incredibly excited to inform you that we started dosing participants in our DYNE-251 program, and our focus today is on sharing an update on our clinical trial progress and details about our clinical trial with DYNE-251. Next slide, please. Dr. Finkel has provided a great overview of Duchenne, so I won't spend time on that. At DYNE, our approach is to develop a best-in-class targeted exon skipping therapy that is better than standard of care and increases dystrophin expression with less frequent dosing to potentially stop or reverse disease progression with our FORCE platform. Next slide. Dr. Finkel introduced our chemistry, and here I'll take a couple of minutes to describe the FORCE platform. Now, this platform drives our efforts to develop and deliver muscle-targeted therapeutics. It's expressly designed to address one major issue in muscle diseases, which is efficiently and reliably delivering antisense oligonucleotides that target the genetic basis of diseases, and targeting those therapeutics to skeletal, cardiac, and smooth muscle. As you can see, DYNE's FORCE platform consists of three components: a proprietary antigen binding fragment, or an antibody fragment, or what we call a Fab, to enable targeted delivery of the antisense to muscle, a clinically validated linker, and the antisense oligonucleotide selected to target the genetic basis of disease. So on this slide, you can see a Y-shaped structure, which is a monoclonal antibody. Well, we chose a portion of this, our Fab, as I just mentioned, because we believe it offers powerful advantages over the larger monoclonal antibodies, including its smaller size, lower overall protein load, and the potential for enhanced tolerability profile. The goal is ultimately to enable targeted, effective delivery of therapeutic molecules to key muscles involved in these disorders. We selected the val-cit linker, shown on the bottom part of the slide, based on its stability and clinically validated safety and efficacy in approved products. Because this linker is used in products that are on the market, we believe this further de-risks our overall FORCE platform. Now, we're utilizing the same Fab and linker across all of our programs. The versatility and modularity of our scientific platform provides flexibility to deploy different kinds of antisense oligonucleotides that are selected to match the target biology for each disease. In the case of Duchenne, where dystrophin restoration is the key, it's critical that we select an exon skipping phosphorodiamidate morpholino oligomer, or PMO chemistry, that targets the specific mutation in the DMD gene. Next slide, please. Now, the mechanism of FORCE is different from other approaches. We're essentially using the cell's natural biology to deliver our therapeutic agent to muscle cells. Now, our Fab binds tightly to something called the transferrin receptor 1, or TFR1. TFR1 receptors are highly expressed across the surface of muscle cells including skeletal muscle, including diaphragm, cardiac muscle, and smooth muscle. Now, the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our therapeutic antisense oligonucleotide to enter those muscle cells to target the genetic basis of disease. So we're able to harness what already exists on the surface of muscle cells to deliver our therapeutic to muscle with this receptor-mediated mechanism. And by targeting muscle specifically by using the TFR1, we have the potential for a wide therapeutic index, resulting in less frequent dosing and chronic administration. Next slide, please. On this slide, we show our FORCE platform in the MDX mouse model, a standard animal model in Duchenne. These MDX mice have a mutation in exon 23. You can see FORCE resulted in robust and durable dystrophin production across key muscles affected by Duchenne with just a single dose of 30 milligrams per kilogram, and we evaluated these mice just 4 weeks later. The top half of this slide shows some of our data demonstrating robust levels of dystrophin expression across key muscles. We were able to achieve high levels of dystrophin expression, approximately 50% in the quadriceps, 90% of the diaphragm, and nearly 80% in the heart. We're extremely excited to see these levels of dystrophin restoration, especially in the heart and diaphragm, where current PMOs have had limitations. On the bottom half of the slide, we show dystrophin localization in the sarcolemma muscle membrane after just a single dose. Now, this localization is important to see to provide confidence in potentially seeing functional benefit. And we saw profound results, including about 70% dystrophin positive fibers in the quadriceps and approximately 80% in the diaphragm and heart at 4 weeks. Next slide, please. Importantly, our FORCE platform has shown to have durability of effect. This slide shows our data on dystrophin positive fibers in the quadriceps out to 12 weeks. So even out to 8 to 12 weeks, we saw sustained localization of dystrophin to the sarcolemma in the quads, as well as other muscles. Next slide, please. Next, we evaluated DYNE-251, our clinical candidate, in non-human primates, or NHP monkeys. Now, these studies are a key step in generating preclinical data before going into clinical trials. We measure exon skipping in the quadriceps, heart, and diaphragm. Please note that in NHPs, we cannot look at dystrophin restoration because these are healthy animals. The key message here is that with our FORCE platform, time on therapy matters. Over a 2-month period, we observed robust exon 51 skipping across muscles. With 5 weekly 30 milligram per kilogram doses, DYNE-251 demonstrated about 52% exon skipping in the diaphragm, 43% exon skipping in the heart, and 18% exon skipping in the quadriceps. Again, we were very pleased to see the level of skipping, especially in the heart and the diaphragm. We also evaluated DYNE-251 in 5 and 13-week toxicology studies in monkeys. Overall, we saw that DYNE-251 had a favorable safety profile with no dose-limiting toxicities observed after 5 weekly doses up to a maximally feasible dose. So we tested these monkeys in these studies at doses much higher than 30 milligrams per kilogram. We're very pleased with the safety profile, which is critical as we advance into the clinic. Next slide, please. So prior to starting our DYNE-251 clinical development program, we proactively obtained expert feedback. We engaged opinion leaders, key opinion leaders or KOLs from various disciplines, including pediatric and adult neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy. Importantly, we have gathered this feedback from experts across the globe, such as Dr. Finkel. Now, KOLs both in the US and outside of the US, including Europe, with experience running early and late stage trials, have provided advice on our overall clinical development program, including our trial design, the patient population, biomarker and functional endpoints, and key safety considerations. And importantly, have helped us to learn from natural history studies. It is clear that KOLs believe dystrophin continues to be a key endpoint for this condition. Now, we've also received advice directly from patients and caregivers, as well as global advocacy leaders, such as those from CureDuchenne and the Duchenne Community Advisory Board, comprising of advocacy leaders from several countries. They provided their insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. And this input has been critical in ensuring a patient-centric design and has included, but it's not limited to, key considerations for choosing a clinical trial, review of and input to our informed consent form, feedback on biopsies and functional outcomes, and identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the process. And based on this extensive feedback, we've developed the proposed the clinical study for DYNE-251 in patients amenable to exon 51 skipping. And I'll start to describe the key elements. Next slide, please. Our clinical trial for DYNE-251 is a global, multiple ascending dose, or MAD, placebo-controlled study with a long-term extension. It's important to note that following the placebo-controlled portion of the study, all patients will be able to receive active treatment. We're enrolling patients with Duchenne who have mutations amenable to exon 51 skipping therapy. Our goal is to enroll approximately 46 male participants at ages 4 to 16 years who are either ambulant or non-ambulant. We plan to evaluate the following outcomes in the DELIVER trial: safety and tolerability, change from baseline in dystrophin protein levels measured by Western blot, muscle function relevant to both ambulatory and non-ambulatory participants. So that would include assessments to help us understand changes in lower and upper limb function, as well as lung function. And importantly, we will be dosing monthly or less frequently in the clinic. With the help of our expert advisors, we're very confident that our study design is robust, will measure key outcomes, and is striving to manage the burden on families. Next slide, please. On this slide, you can see some additional details, including the trial design. I won't go into too much detail here, but rather highlight a few points. Again, as I mentioned, this is a multiple ascending dose study in patients who are amenable to exon 51 skipping that include both ambulatory and non-ambulatory patients. In the United States, we have 7 cohorts starting at 0.7 milligrams per kilogram up to 40 milligrams per kilogram of the PMO equivalent, with a 2-to-1 randomization in the first 5 cohorts and 3-to-1 in the last 2. Now, we require IV administration of DYNE-251 or placebo every 4 weeks. We also require 2 muscle biopsies, one at baseline and another at 24 weeks, starting in the 2.8 mg per kg and higher cohorts. So no biopsies are required in the first 2 cohorts. And importantly, patients in the placebo-controlled period are escalated to the highest tolerable dose in the open label extension and the long-term extension. So all patients are guaranteed to be on active drug after the 24-week placebo period. And this global trial is designed to be registrational, and we expect to have safety, tolerability, and dystrophin data in the second half of 2023. I'm also very pleased to announce that we've recently received FDA Fast Track designation for DYNE-251, and this provides us an opportunity to expedite the development of DYNE-251. Specifically, the FDA grants Fast Track designation to facilitate the development and expedite the review of drugs to treat serious conditions and fill unmet medical needs, with the ultimate goal of getting important new drugs to patients earlier. And we're very thankful to the agency for their continued partnership with us. Next slide. And finally, we have tried to take every opportunity to listen to the Duchenne community on their needs for next generation treatments. We're working urgently to deliver for the community, including families and healthcare providers, to bring a potentially transformative treatment forward. Thank you to the families for being generous with your thoughts, advice, and for the sacrifices you continue to make. On behalf of DYNE, I would like to thank you all for your time and attention to listen to this presentation. and I'm pleased to take any questions. Thank you.
Ash, fantastic. That was a real tour de force, with no pun intended there. It was great to see all of that data come together. I got to say that, you know, I've seen some of the data before, and it's an impressive collection of preclinical data, particularly the single dose in the mouse, where you're seeing a very long pharmacodynamic effect. And it's also particularly impressive to me looking at the NHP data, where these are not dystrophic muscle cells, you know, and being able to get that amount of skipping going on, you know, really speaks, I think, to the power behind the platform in here. Could you just comment on how that's guided the dosing regimen that you're using in the clinic in patients?
Sure. Thank you. Thanks, Mike. Yeah, it's a great question. So, you know, one of the things that we've certainly heard from the community is for those therapies that are on the marketplace right now in the US, which is fantastic, the weekly dosing does provide a huge burden. And so when we were developing DYNE-251 and looking at this within the preclinical studies, we were able to derive from the data the knowledge around the durability of effect. And so with just single doses, both from low as 10 milligrams per kilogram all the way up to 30 milligrams per kilogram, we were able to show very good durability of effect. And then you saw the data in the 30 milligram per kilogram single dose arm experiment that in the quadriceps. And then the data that I did not show here, we also have data both in the diaphragm and the heart that shows durability out to 8 to 12 weeks. And so this, along with the NHP data that I showed you, gives us a lot of confidence that the most frequent we will need to dose in the clinic is monthly. And we actually believe there's the potential to dose less frequently than monthly dosing as well.
Yeah, I suspect that there myself, actually. It will be interesting as you look at a range of ages of patients. Those older patients with less muscle may require more rapid dosing or more frequent dosing than, say, younger patients with much more muscle, you know, where you're able to build up the amount of drug that you've got on board in there. It'll be a very interesting clinical trial to tease that information out.
Yeah, absolutely. And I do think that, we have the potential to really stem the cardiopulmonary decline. So, although that decline starts very early, even as early as early as age 6, we recognize that in those older participants and the older boys, the ability to stem any additional cardiopulmonary decline is going to be really critical. And so we're seeing the ability to get into those muscles, unlike any other modalities that have been out there so far.
Yeah, you actually got to my next point. You and I have talked about this over the last few years, but I've always been struck by the cardiac targeting that you see here. And it really is a very important component of the overall target product profile. It's something that's not met in some of the earlier chemistry and is sorely needed. And I look forward to seeing this play out longitudinally as we look at cardiac function over a few years of treatment.
Yeah, and I did, so thank you, Mike. And sorry, I did maybe a couple of things. So one, that speaks to the utility of using the transferrin receptor. The TFR receptor is highly expressed on the cardiac muscle. And so developing this Fab that's engineered to bind very tightly to that TFR1 receptor provides a really good mechanism for us to be able to drive the ASO into cardiac muscle. And then coming into the clinical trial, we do have both echo and cardiac MRI that we are also utilizing within our clinical trial.
Yeah, super. My next question really was related to that, because there's real differentiation in this platform, because of what we see targeting both diaphragm and particularly cardiac muscle. Could you speak to that? Because I feel that's an important component for families to know. There's a number of choices today for clinical trials, and having people really recognize some of the differences that exist out there really is an important component of what this DYNE compound does.
Sure. Do you want me to take that, or do you want to have Dr. Finkel take that?
Your choice.
All right. I'm happy to go first here, since I'm off mute, and then I'm going to hand it over to Dr. Finkel, if that's okay.
Sure.
All right. Yeah. So from our perspective, this offers offers the community what's definitely needed in the sense of another way to attack Duchenne. We know that there are many modalities that are out there that are in development, and hopefully all of them make it to market. Hopefully all of them come to fruition, because we believe that the SMA market, provides a bit of an analog here, that to be able to really fight and battle Duchenne, it's going to be a multimodality approach, small molecules, exon skipping, and gene therapy. And so we believe that it's going to be we're going to have to attack Duchenne in a variety of ways. In terms of some of the specific differences, we've talked about the unconjugated PMOs that are delivered. There is not a mechanism that's driving them into the, actively driving the ASOs into the muscle cells, into the nucleus specifically. Then there are the PPMOs, or the cell penetrating peptides, the CPPs that are attached to the PMOs. And there are compounds that are in clinical development from various companies right now. And so there is emerging clinical data with those modalities. And again, they do appear to be able to penetrate muscle cells. There also appears to be some advantages and potentially disadvantages in terms of the side effect profile that's emerging with the PPMOs. And then, of course, gene transfer therapy, it's AAV mediated, where there is a choice that companies are making in terms of the kind of, the elements of the DMD gene that they incorporate into the capsid, which is not, you're not able to take the entire DMD gene and put it into the capsid. So companies have to make different choices. So essentially, you're creating a micro dystrophin or a mini dystrophin that's exogenous, that's sort of not native. And so again, there's emerging clinical data with that modality, as well as some uncertainty around the benefits of microdystrophin versus near full length dystrophin. The durability isn't quite understood yet, and of course there have been some safety issues with respect to the capsid, immunogenicity concerns, as well as transgene concerns. And so those are some of the ways to think about the differences between the FORCE platform versus others. And then, as I said, what we're doing is a receptor-mediated delivery of the ASO using the sort of the cell's natural biology with the TFR receptor to drive the PMO into the nucleus of the muscle cell.
Thank you. Dr. Finkel, did you have a comment to add?
Oh, I would just amplify, Mike, that I've been particularly impressed at how well the DYNE-251 molecule gets into diaphragm and heart tissue, perhaps even better than skeletal muscle. Now, we need to see, is that true in dystrophic muscle in our little boys and young men with DMD? But I think the protocol that's been constructed and has now initiated will answer some of those questions. I think it's been carefully constructed to both learn how every 4-week dosing will do. We have the muscle biopsies to be able to identify the dystrophin expression. We're capturing what I think are the main clinical features that we want to look at to try to identify the clinical response. So while this is an early phase 1, phase 2 study, it has all the features that I think will allow it to support regulatory approval in due course. And I'm very excited to see how this moves forward.
Yeah, thank you for that. My own view is that exon skipping will continue to be a cornerstone of therapy going forward. We're producing a fairly large protein here, something that's 95 or more percent of the length of the full length protein, and one that we typically associated with an asymptomatic Becker disease. So it really is an important protein to put in these sorts of quantities. One other thing I'm going to move on to the clinical in a second, but I just wanted to, while I'm talking to you, Dr. Finkel, I was struck by your life cycle slide. And it's clear, that in very young infants and children, the disease is going on unrelenting, even though it may not be apparent to the outside world. Where do you see the importance of continuing to get into younger and younger children with these types of therapies?
Right. So a very important question, and I think it remains to be seen. when is the optimum time to initiate treatment in a boy with DMD? As you know, there's been a recent request, an application to add DMD to the newborn screening panel.
Mhm.
And that's because there are therapeutic options that are now evolving that would allow us to interrogate that, to say, okay, we've identified a little baby who has DMD, and now what do we do? And that's really the question, is when to start steroids. Well, none of us like steroids, even though we know that they work, and we certainly don't want to start them in a little newborn baby. If we had what we call a targeted treatment strategy that works at the level of the fundamental cause, meaning the dystrophin deficiency in these boys. And as I showed in that earlier slide, we know that under the surface, there is activity going on. The disease process has already taken hold. So to me, if there was a safe drug that can restore dystrophin and stabilize these muscle fibers, particularly during early growth and development, because as we know, little babies, they double their weight pretty quickly by a year, and a lot of that is muscle. So I think that first year is even particularly important, because most of the boys don't have symptom onset until about age 2.5. They're not diagnosed till typically 4.5. So the whole idea of newborn screening and identifying these babies, what we might call pre-symptomatically, gives us a whole new window of opportunity. Similarly, I think in the boys who are already symptomatic, you could see from that early slide that what's happening in the muscle is a relentless progression, but being able to stabilize that and perhaps even reverse some of that as new muscle fibers are populating, what we call this from the satellite cells, I think gives us an opportunity potentially to even see some improvement. The current exon skipping drugs do slow down the decline, but it's not something that I can see visually in the clinic. It's something more that we measure statistically. But I think with this next generation of ASOs like DYNE-251, I'm keeping my fingers and toes crossed that we may actually see some improvement in these boys if we treat them early enough. And then there's that third group, which are the boys who have more advanced disease, young men, where we really need to get the drug into the diaphragm and the heart. So maybe we have to have different expectations, in other words, based on the different stage of disease that we're treating. But if we can stabilize or potentially even reverse the cardiac and pulmonary side, that would be a game changer. And it would really improve quality of life as well. So I think DYNE-251, in my mind, has the potential to really be applied in all three of those phases: the pre-symptomatic, the early symptomatic, and the patient with more advanced disease.
Well, thank you for that. You're speaking to the choir. We believe exactly the same thing. I also feel that the way the trial is set up, taking a broad range of ages, is going to give us some insight into some of the answers in there that's going to be useful actually guiding future studies. Aisha, if I may move on to the clinical trial,
Sure.
Could you just say a few words about inclusion-exclusion criteria, and in particular for patients who may be on an Exon 51 skipping drug and want to move over? What is the washout? How is the company thinking about that?
Yeah, no, happy to. So maybe just at a very high level, some of the key criteria. We've talked about the amenability to exon 51 skipping, 4 to 16 years of age, ambulant, non-ambulant. I will say, in terms of the prior use of exon skipping therapy, we do allow patients who have been on prior exon skipping therapy, and we require a 12-week washout.
Okay.
And so we know that is not insignificant, and we know that's going to be a very important consideration for any family. But we did want to be able to provide the opportunity to be able to have families who have been on a prior exon skipping therapy, and yet make sure that we maintain the scientific integrity of the study with that washout period. And that washout period is no different than what other next-gen studies have, next-generation therapeutics have had in their clinical trials as well. So it's a 12-week washout. I will mention also.
Can I add just a point, Ash? Because I really strongly support this, and at our institution, as we've reviewed it with our ethics committee and everything, we've had to defend this, and I think there's a strong rationale to support this, and the families seem to be endorsing this as well. They understand there's a risk, but I think it's a well-designed study, and I think we're going to be watching these patients carefully and making sure there's not undue decline, and that the opportunity here, if you balance everything, I think the benefit for these patients really, is something that we can certainly balance with that small risk over that short period of time, that 12 weeks.
Oh, that's great. Thank you for that. I was just going to add that we do exclude any participants who have received gene therapy. While we believe it's going to be a multimodality approach in the marketplace, given that this is a phase 1/2 first-in-human study, we could not include patients who've been on gene therapy.
And if I may ask, patients that were on vamorolone, for example, given steroid background?
Yeah, it's a great question. We get that question quite a bit. So currently, as Dr. Finkel alluded to, vamorolone is not yet approved by the FDA. So it is still considered an investigational product. So we would require patients washing off of vamorolone prior to entering our study. We do require that patients have a stable dosage of glucocorticoids, such as prednisone, deflazacort, for at least 12 weeks prior to starting the study, starting DYNE-251. But we cannot include patients who are on vamorolone at this point.
Okay, thanks for that. You actually answered part B of the question, which was the washout time and the stability on the switchover. I have to say, no one ever likes to give biopsies or see them, but I congratulate you guys on only having 2. Could you just talk about the biopsy, whether it's an open a needle biopsy? Is there a choice in there? Is that done at the clinical center?
Yeah, great question. Again, and Molly can speak to this, I'm sure, quite a bit. We know that the biopsies are an important consideration for families. We are doing open biopsies, not needle biopsies, so we require open biopsies. It'll be done at the clinical center. The reason for that is we've learned from previous companies' trials that to ensure that we are getting muscle that can be evaluated for exon skipping, for dystrophin restoration, for dystrophin positive fibers, to be able to make sure that we are getting enough viable tissue. The best approach is an open biopsy where the surgeon or the neurologist can actually look and visualize the muscle. So that's why we're doing it. We've learned a lot from previous studies.
Okay, thanks for that. I know I can find a lot of information on clinicaltrials.gov. Can you just give a little bit more information out about the number of sites you plan to have, the number that are active right now?
Sure. So we have a couple of active sites right now that are posted onto our clinical trials website posting. So we plan to open up several more sites in the US, many more sites. And of course, we're going to some of the most experienced centers that have done Duchenne trials, such as St. Jude, where Dr. Finkel is. As these sites get activated, we will post them on the clinicaltrials.gov website. And so stay tuned, and you can visit the website for updates. We update frequently. So right now, I can't tell you exactly how many sites. We're just getting them up and running and activated, but we will continually update on our website.
Okay, and I know that Carrie is in the background listening. As you do that, we'll certainly get that information out to the community through CureDuchenne Cares, etc.
We
have a number of international sites as well that will be opening up. So it's a global trial.
And that's
I was just going to ask Molly.
in Europe.
I was just going to ask Molly to comment on biopsies. Anything else on the biopsy considerations or other trial considerations for families, Molly?
I wouldn't add anything except what you all know well. These parents are sophisticated, educated parents, and they know why these biopsies are needed. And I think what we've heard in the patient advisory workshops that we've held is just, please do the right biopsy to collect the right amount of tissue so that it's not wasted.
Yeah, totally. Molly, could you just add a little bit more color onto the ex-US sites, whether Canada, Europe, the United Kingdom, for example?
No.
Okay.
I'll throw that back to Ash.
Sure.
All right.
Yeah. So Molly's absolutely right. We are, we're doing a global clinical, it's a global clinical trial. We're going to have several sites outside of the United States as well. Whereas I'm sure you all know, there are no approved exon skipping therapies, for example, in Europe, just in a select few countries. So we are in the process of engaging with those sites outside of the US. And again, as they get up and running and become active, we will be sure to post them on the website. So right now we don't want to provide any specifics, but as we activate them, we'll post them.
Okay, super. My final one really is it's a question from me. I tend to ask questions for the community, but this one, you're bringing in a bunch of patients that are covering a broad age range. How do you actually set them up so that you can, the signal or the endpoints may be different as you cross through those patients? How are you setting that up, or how are you randomizing those?
So the way that we are running this particular clinical trials, as I said, we have a 2-to-1 randomization in those first 5 cohorts, 3-to-1 in the last 2. And we obviously wanted to serve the non-ambulant families because they've been so underserved. So we are allowing at least 2 non-ambulant patients per cohort. And that's the way we're trying to sort of balance out this particular trial. We also are not, we also want to be prudent of how long a family or a patient has been in the non-ambulatory status. And so they cannot be non-ambulatory for more than a couple of years. And so that's the way we're kind of balancing things out. So hopefully that answers your question.
It does. And I just want to add my personal comments in there. Like I want to say thank you for that. It's not often that people really think about the broader population out there. And there'll be a lot of parents that I will talk to that really will be happy to see the inclusion of a broad age range in this. So thank you very much. I'm going to wrap up. The thing that I would say is real congratulations on opening up the study. I think the preclinical work has got a lot of people very excited, and hopefully that'll roll over into recruitment and that'll feed. And again, congratulations on, the recent fast track designation from the FDA. I think you guys are in a great position, and I hope that we can help get this message out about where you stand today. So thank you, Ash. Thank you, all of you guys, for participating.
Thank you so much. Thanks for having us.
Carrie, over to you.
Yeah, Mike, you said I just want to express my gratitude to DYNE and to Dr. Finkel today. Whoever is watching this fireside chat, we hope that you learned a lot. But if you do have questions still, or if you have specific questions regarding your family, maybe your child's mutation, your specific situation, please do reach out to CureDuchenne. It's CARES, C-A-R-E-S, at CureDuchenne.org. And we collaborate often, with people like Dr. Finkel, with DYNE, to make sure that you're getting the whole picture and getting the correct information to guide the decisions that you need to make. So Mike and I are very happy to take on those questions, to meet with you and your family. And I think that's our kind of closeout of the fireside chat today. Thanks everybody for being here.
Okay, super job.